having me here. I'm sorry I couldn't be in person. I would have loved to be there, but I just had another conference at the same time. So the work I'm going to talk about is QuCo, Quartet-based co-estimation. It's all the work of my student. Oh, I'm sorry. My student Mariam, who has graduated since then. So I'm going to present instead of her. So the context is gene tree, species tree discordance. Everyone in this audience knows, right? You have gene trees that are discordant from the species tree for various reasons. What we are going to focus on today is ILS, which is this process that takes multi-species coalescent into a species tree context, so you can have lineages where if you kind of follow the coalescence of these lineages back in time, if they don't coalesce in the common ancestor, you can get incomplete lineage sorting. That's what we will be focusing on. The model that we use to study multi-species coalescent is ILS is called multi-species coalescent. And what really matters under this model is just a very simple observation. What I am going to use today, which is the model defines a unique distribution over topologies of the gene trees if you give me the species tree with branch lengths in coalescent units. And in particular, I'm going to use one fact, only pretty much just this one fact from the model, which is if you have 4 species, a quartet, and you give me the topology and you give me the length of this internal branch in coalescent unit, the probability of the 3 species trees, 3 possible quartet topologies are known. It's really easy to kind of show that these are the probabilities. Say I missed the citation here. This was shown in Allman et al. 2010, but I think the earlier papers, even back in 80s, something similar to this was known. Okay, so that's multi-species coalescent. And a little bit broadly, we have a kind of a hierarchical model that we assume. We assume the species tree through a model generates your gene tree. So there's a distribution over gene trees. Each of those gene trees, again, through a model of sequence evolution, generates your sequence data, right? And the model for the gene tree evolution, like I said, is multi-species coalescent. For sequence evolution, could be anything like GTR, for example. Now, this is how data is generated at the time of inference. Things kind of, you can do it in different ways. The simplest approach that first would come to your mind is, well, let's take each gene, use one of those models, infer a gene tree. Now you have a collection of gene trees. And perhaps we can then summarize those gene trees to get a species tree. And that's kind of one of the dominant approaches in the field. Is we call it two-step or summary-based methods. One of the methods for this summarization is Astral, which is something I presented in ISMB, I don't know, maybe 8 or 9 years ago. I don't remember. Okay, so this is one approach. This is not the only approach. One alternative is co-estimation. And co-estimation is when you try to do this inference of gene trees and the species trees all together. Now, the question is, why would you want to do everything together, right? And the answer is the issue of gene tree estimation error. So the heterogeneity that we observe in the gene trees that we estimate from data, they can have 2 reasons. They can have various biological reasons like ILS. That's one thing. But they can also have noise. When you infer gene trees, they have error, and that error adds to the discordance. So when you run a species tree estimation methods like Astral or other ones, no matter which one you try, if you do simulations and you control the amount of gene tree error, like we have done, for example, here, this is from a much earlier paper, you will see that as the amount of gene tree error goes up, the amount of species tree error also goes up. And this has been documented again and again and again. One of the earlier ones was this paper from Ed's lab, but now it's a very well-established thing. There is also even theory suggesting as much. So gene tree estimation error is a problem. There is this idea that co-estimation can help with that because you are estimating all gene trees together. Now, there are methods to do co-estimation. There are quite a few of them out there. The most famous one is Star-based. But the issue is limited scalability. You can barely run it on I don't know, more than 50 species or something like that, right? So you can run it on thousands of species or even hundreds for that matter. And the reason is not that these are not good software. It's just that they are trying to solve a very difficult problem. They are trying to sample the posterior distribution of, so when I say theta, I mean the species tree, the posterior distribution of species trees and all the gene trees at the same time. And both the species tree and gene tree have discrete parameters like their topology. They have continuous parameter. There is like a huge space that you are trying to sample just using MCMC. And so it's not so surprising that the methods don't scale. It's a very difficult problem they are trying to solve. So what do we do? In this paper, there have been various attempts in ISMB, I think 2019 or 20, Nachler's group had some alternative approach. Various people have been thinking about how do we do it in a somewhat more heuristic manner, but in a way that is scalable. And the paper I present today is in that way. So what we do is, first of all, we work on quartets. So if you have n species, you can take either all the n choose 4 quartets if n is small enough, or you can select the subset of quartets. There are various ways to select subsets of quartets so that with a much fewer number of quartets, you can still kind of reconstruct the species tree. So either way, you select enough quartets to be able to reconstruct the tree. For each quartet, we are going to infer a species tree for that quartet with its branch length. And then at the end, you can, of course, combine these quartets. So that's the general idea. Now, for each quartet, so from here on, I'm going to focus on one quartet. Let's discuss what we do. We have a parameter theta, which is what we are interested in. It's the species tree. It has the topology, let's call it T, and the length of this internal branching coalescing unit, which is D, or sequence data, or S. I am not terribly interested today in gene trees. I'm mostly interested in the species tree. And what I want to note first is that instead of co-estimation, we can do marginalization, right? So we can try to kind of look at the marginal probability of the sequences given a species tree, where you marginalize over the gene tree distribution. So here you have the probability of your sequence data given genes and the probability of gene trees given the sequence data. Now I have an integral here because the space of all gene trees is a continuous space because you have branch lengths. And by marginalization, maybe we can make the problem a little bit easier because we are not trying to get the right distribution for all the gene trees at the very kind of fine grain. But even this is a difficult thing to work with, even if you're just working on one quartet, because you have these integrals, right? You have integrals over all these continuous parameters. So we actually go one step further to make the problem a little bit easier. We work on topology. So you could also marginalize over topologies, right? You can marginalize a variable over whatever you want. So we marginalize over all possible gene tree topologies. And so now instead of an integral, we have a sum. The number of things that we need to sum over here is not super small, right? You have 3 topologies possible for each gene. So you have 3 to the k possible gene tree topologies in total. But there are some nice things that start to happen. So for example, the probability of gene trees given the species tree, you can easily compute it analytically using those little equations that I showed earlier. And this is something that looks familiar to you guys. This is sequence likelihood, right? So we are starting to get terms that we are a little bit more comfortable with. Okay, so let's keep going. That's the equation I had. I want to keep simplifying it. This probability over all the set of all genes given the species tree by conditional independence of the gene trees, you can just write it as a product. So that's good. And of course, these equations I showed earlier, right? The probability of the gene tree given the species tree, we have these simple equations for it. Okay, so now let's focus on the likelihood of the species tree. Now, and so sorry, not the species tree, the likelihood of the sequence data. So the first term here. So for this first term, I haven't written it as a product yet, because you can't actually, if the gene trees, because the gene trees here are topologies and not gene trees and branch lengths, you cannot immediately write it as a product. But if you're willing to make enough assumptions, you can, right? And if we are in the business of trying to get something scalable, so when we need to make assumptions, we just make assumptions. So here we had to make some assumptions that are a little bit on the heavy side. They basically boil down to assumptions about independence of branch lengths from topologies, which do not hold under many models of branch length, if you think about branch lengths. But let's make those assumptions. Once you make that assumption, you can write this product also as a product over gene trees, right? So now our equation looks a little bit better. And in particular, this probability in the middle is, or just you have one gene tree and you're trying to compute its posterior probability, right? And so this is much easier to work with. Here, I'm not writing equal, I'm writing proportional too, because I went from the, I'm kind of flipped the order. So you have to also make some assumptions about the prior. So there are a bunch of assumptions going. This step is the most tricky one instead of it being correct. Anyway, once you get to this equation, things become much nicer, because the posterior probability of gene tree topologies are really easy to compute, in the sense that the tools exist, right? So you have your input sequence data. You can run any MCMC sampling method that you like, for example, like Mr. Bayes or any of those. They give you an MCMC chain of gene trees with different branch lengths and topologies. We are marginalizing over branch lengths. We don't care about branch lengths. So you just simply count how many times you saw each of the three topologies, right? And that gives you the marginal probability of the topologies for the sequence data that you have, marginalizing over branch lengths. And you have a quartet, so this is easy enough to do. And you can write these in a vector of length 3. We are going to call it P112131, right? So this is for the first gene. And then you have k different genes, right? I'm going to use k for the number of genes always. You have k different genes. You do the same exact process. You run MCMC for all of them. You get these probabilities, right? So for each gene, you have a distribution over the topologies. And you can take all of these and put it in a matrix. So that's the input to our algorithm. That's all that we are going to need to know from the gene trees. Basically a 3 by k matrix where each column adds up to 1, because each gene must have one of the 3 topologies, right? Okay, so we have this matrix P, and I just rewrote the probability of the genes, the probability of each gene tree given the sequence data is posterior. I replaced it with this, with elements of this matrix P. I didn't do anything fancy here. And I just, so now you have something that looks a little bit nicer. And then there is a very nice trick. So what's not so nice here is that you have a product inside the sum, but this is a very specific kind of sum and product. And it turns out that you can kind of rewrite this equation in a way where the product is out and the sum is in. There is no approximation here, nothing. This is just like simple algebra. So that's a very nice trick. And it takes us to an equation that looks incredibly nice. So the probability of your sequence data given the species tree quartet becomes just this product over all the genes of this little equation. And once you have that, once you have just one product, everything is nice, right? Because you take the log of the probability, you get your likelihood, becomes a sum, and now we have the likelihood function that we can easily optimize. So this is my likelihood function. Again, the input was generated with Mr. Bayes, right, or whatever Bayesian method. You get this matrix. You for each quartet, you have 3 topologies. For each of the 3 topologies, this would be their likelihood. Now, of course, we don't know what the branch length is, but we can optimize for it. And it turns out optimizing for D in this equation is relatively easy. I don't think we can do it analytically. I haven't figured out how to do it analytically, but the numerical optimization has all sorts of nice parameters, so we can easily optimize and find the maximum likelihood. So say I'm missing an argmax here, so all of these should have an argmax in front of them. So you have 3 topologies. For each of them, you maximize the likelihood, you find D, but also the likelihood, and you can pick whichever one gives you the maximum likelihood as the topology, right? So that's all there is to it. For if you have only one quartet, that's the method. Let's look at some simulations. So to be able to study these in challenging conditions, what we do is we do simulations under sort of Felsenstein zone-like conditions. So, or a species tree has a branch length D in the middle. We are simulating quartets. We change D, so very high ILS to moderate levels of ILS. But we also came up with a procedure. It's not trivial, but you can figure out the mass so that the expected branch lengths of the gene trees have this Felsenstein zone-like pattern where you have 3 short branches and 2 long branches, and you can control what these long and short branches are in expectation. So you can create gene trees that have a lot of error, ranging from 15% all the way to 70%. Okay, so what makes the problem difficult is if these long branches, lambda, are a lot longer than the short branches, mu. So we kind of looked at a grid of various lambda and mu values. We have more data in the paper. I'm showing a subset here. And we are comparing QuCo. So on the y-axis is we do 20 replicates of these simulations. How often do we find the correct tree, right? And on the dotted line is what you would get if you just use Astral. So basically, the most frequent maximum posterior gene tree. The solid line is the QuCo. So that area in between is the improvement you get going from Astral to QuCo for all of these conditions. So some patterns, there are some conditions we get a lot of improvements. There are some of these difficult conditions where the long branch is 20 times longer than the short branch or something like that. In those conditions, neither method works. There's just too much long branch attraction for either method. And there are, of course, conditions where both of them work pretty well. You could, of course, if you reduce the amount of ILS, the problem becomes easier for both methods, and some of these difficult conditions get rescued, but you can still have conditions where neither method works well if you have enough long branch attraction. Okay, so that's comparison to Astral. Now, this kind of method that I described takes as input Mr. Bayes distributions independently estimated and kind of combines them together, right? And there is another method called Bucky that does the same thing. This is from Cecil Anne's group from back in 2010. And when we compare QuCo to Bucky, the improvements are much less. In fact, there are a couple of cases where it's less accurate. So, for example, here Bucky is more accurate than Astral, than QuCo. So the improvements compared to this similar method that takes into account gene tree uncertainty are less, but overall there is still improvement. Okay, so that's about topological. So let's think about branch length accuracy. Oops, sorry, my branch length accuracy method figure for some reason doesn't show. In terms of branch length, I'll just say, I apologize, I don't know why it doesn't appear. The branch length has become a lot more accurate than Astral. Astral branch lengths are known to be biased, underestimated. The QuCo branch lengths are not. The next thing is gene tree estimation error. So Once you get a better species tree, you can go back and update the posterior probabilities of the gene trees. That's why we can call the method co-estimation. However, when we look at the error in the gene trees, so here on the y-axis, I'm showing the improvements in the gene trees going from astral to QuCo, or not from astral, from not doing co-estimation to doing co-estimation on the y-axis. And on the x-axis, I'm showing the improvements in the species tree. The bottom line is this: under conditions where the species tree improves, we do often get improvements in the gene tree. But under conditions where the species tree doesn't change, sometimes you get improvements, sometimes you can even get gene trees that are worse. If your species tree is incorrect, it can make your gene tree estimation worse. So that's important to keep in mind. It's not always better for your gene trees. Okay, so what do you do if you have more than 4 species? Like I said, you can select subset of species, run QuCo on each, and then combine. That combination step at the end is something that is very well studied. You can use Astral itself, or you can use methods like QMC that are designed for it. Okay, so we are going to look at some result. Oh, again, my results don't show for some reason. I'm sorry, I don't know what's happening with this. Some of the results are not showing. So let me just, the results are all in the paper, but the bottom line is that if you have 30 species, we can run it on 30 species or even 100 species. 30 species, you don't subsample quartets, and 100 species, you do subsample them. In both cases, we see some improvements compared to running Astral on gene trees. The improvements are not quite as dramatic as if you had those quartet simulations that I showed before, but there is still some improvement. And again, I apologize. I refer you back to the paper for the figures. So to summarize, QuCo finds a maximum likelihood quartet species tree for each quartet, and then for more than 4, you have to kind of heuristically run it on a bunch of the quartets and then combine them. It can improve the species tree dramatically in simulations under the most difficult conditions, but not always. Sorry, not most difficult, under some difficult conditions, but not always. The impact on gene tree accuracy is mixed. Sometimes it improves it, sometimes it doesn't. And then there are a lot of things that we can do in near future. For example, instead of doing this, run it on each quartet separately and combine trees, we can kind of search for the full tree using a composite likelihood, pseudo likelihood kind of approach. We can think about better ways of selecting quartets, and we can go from quartets to quintets, right? The only thing we need is the distribution of gene trees. I think those are known for quintets, so the approach in principle can extend. So with that, let me just remind you, this is the work of my student Mariam, and let me acknowledge all our sources of funding to do all the computation and the work and the stuff, and perhaps take some questions. Hello.
Hi. So I think we have about 5 minutes for questions. Are there any online questions? So I'll ask a question then, which is it possible, instead of using Mr. Bayes or some Bayesian method, do you think it would be valid to use bootstraps? Obviously the Bayesian interpretation of the bootstrap is a little dicey, but what would you think about that?
That's a very good question. And well, unfortunately, one of my slides that didn't show was something similar to that idea. So, of course, like if you go to Bootstrap or things that are even simpler, the theory might start to kind of not hold quite as much. But then again, here we are more empirically oriented in this kind of thing. There are a lot of assumptions and simplifications anyway. So maybe we can do that. So when I talked about this is one of those slides that didn't show. Here, what we are working on are these 30 taxon trees, and instead of running Mr. Bayes, we just run IQ3. And with IQ3, you can get, for each branch, you can get, in fact, what it can give you is the set of likelihoods for all the quartets. So it can give you marginal likelihood of quartets for the three topologies for all the quartets. So it's not quite bootstrap, but it's not Bayesian either, right? And the method worked pretty well there, right? So we are comparing Astral run on IQ3 maximum likelihood gene trees, as opposed to QuCo and these like quartet frequencies that IQ3 can, or I guess quartet distributions that IQ3 can estimate. And they work a little bit better. We haven't tested bootstrapping per se. Maybe we should. The challenge is going to be, I think we're going to have too many zeros, and in bootstrap you might not see some topologies at all. Whereas with these long MCMC chains, you see most things at least once. So if we do enough bootstrapping, maybe that would work.
I suppose you could also add pseudo counts to get rid of the ones.
Yeah, and actually we don't even exactly have, like, the method can handle zeros, but I'm just saying that those zeros might make the estimates less accurate. Now, if there was, you could also add zeros in ways that correspond to a prior distribution, and if maybe there is a clever way to do that, that would work. We haven't quite explored that.